Universal Protein Resource, or UniProt, is a database that contains extensive information on protein sequences and annotations. It is curated by the UniProt Consortium, which is a collaboration of the European Bioinformatics Institute, the Swiss Institute of Bioinformatics, and the Protein Information Resource.
UniProt KB is a central database of protein sequences with annotations and functional information. It consists of SwissProt and Tremble. SwissProt derive from publications reporting new sequence and review articles to facilitate the periodic revision of groups of proteins, while Tremble is supporting the SwissProt protein sequence databank with computational annotations.
So for the next component of UniProt, we have UniProt, where it stores all publicly available protein sequence data in a single, stable, non-redundant collection. Each unique sequence is assigned a UniProt identification, and it provides an incremented each time the underlying sequence changes. Next is we have UniRef, where it provides non-redundant UniProt data collections to span sequence space at various resolution. For UniMesh, it focuses more to store sequences from environmental samples.
Welcome to UniProt. These are the data display available in UniProt, UniRef, UniPark, and Proteomes, and also the supporting data elements supported by the UniProt. At the right hand side, we can see the latest news available, and also we can download latest release here. At the search bar, we can click the button advanced for advanced search, but for our example, we just simply use IGF1R and then click search. The result shows name of organism available, protein names, and the entries. You can filter by review, there are review paper, also the popular organism. There's few features available in UniProt, which is plus, align, retrieve ID mapping, peptide search, and sparkle. And the result obtained can be in form of function, protein reaction, 3D structure, sequence, and location.
After that, FASTA format can be obtained by clicking the format and then select FASTA. You can see the FASTA format is displayed. Then, multiple sequence alignment can be done through UniProt KB by selecting two or more entries within the same gene names. The result will be displayed after a few seconds. From the sequence given, desired annotation can be done by clicking the left side boxes, for example, the domain, helix, number of turns, and etc. That is all for UniProt demonstration.
Now, we will do an evaluation between UniProt with another protein databases. The protein database chosen here is the Protein Data Bank, PDB. UniProt is a sequence and annotation for protein databases, while PDB is essentially for protein 3D structure generated using X-ray crystallography or NMR spectroscopy. From the comparison of interface, UniProt has a simple interface for protein searching, where the search engine is located at the middle of the interface. UniProt also provides different categories of documentation such as the proteins, the species, the protein clusters, and the sequence achieved, and also different analysis tools such as BLAST, Align, search with list map IDs, and search peptides. For PDB, it provides a more complex search interface than UniProt, as it has its search engine located at the top right of the interface. PDB also does not provide the categories of documentation for protein information, where a user might need extra steps to locate for information. There are also no analysis tools found in PDB as in UniProt. From UniProt, as we can see in the entry summary page, there is more information categorized by UniProt at the side of the database interface for easier and clearer information searching. For example, for Protein Data Bank, from the entry summary page, we can see that it provides less information about the protein search at the top side of the database interface. Furthermore, the data in UniProt provides data confirmation about the protein search, indicating a high-quality, manually annotated, and non-redundant protein sequence database that brings together the experimental results, computed features, and the scientific conclusion. As for PDB, there are no symbols indicating whether the data are reviewed or not, which may lead to problems such as the redundant of protein sequence database.
UniProt provides the 3D structure of the protein in black and white color, but color and description will appear as the arrow point on a particular part of the structure. PDB provides the 2D structure of the protein, and the 3D view of the structure can only be accessed by click and open the 3D view structure hyperlink. There will be colorful 3D structure of protein and the description aside. UniProt provides the detail of protein, for example the FASTA format sequence in this website page together. PDB have less information in the website page. More detail, for example, the FASTA format sequence of protein can only be accessed by download file.